First fact: genes do not issue direct commands, but rather steer life through the fine direction of expression. Inside the cell, the same 20,000 plus protein-coding genes are read differently. In a neuron, certain pages are open. In a liver cell, entirely different ones. And in a stem cell, almost the entire script remains closed for now. At the control panel are promoters, enhancers, and silencers. DNA segments to which regulatory proteins attach and decide how loudly a gene sounds. The majority of the genome consists of such regulatory fields, not instructions for proteins. And it is they that set the rhythm of development, response to stress, and adaptation to diet and diseases. On top of DNA lies epigenetics. Methyl labels and chemical modifications of histones change access to genes without altering the text. So the same segment can remain silent in childhood and speak in adulthood, or vice versa. Add imprinting, where one of the parental alleles is intentionally silenced, and random X chromosome inactivation in women, which makes the organism a mosaic of X-linked traits. Even hard mutations work probabilistically. There are the concepts of penetrance and expressivity. A gene increases risk or sets a range of manifestations rather than forging destiny once and for all. Therefore, what is inherited is not a ready-made sentence, but a system of settings and tolerances that trigger differently in different tissues, ages, and environments. Life is a constant work of switches. When cells divide, wounds heal, sleep changes, a muscle grows after training, it's not that a gene turned on, but hundreds of coordinated turn-ons and turn-offs, timed, located, and powered. In this sense, it is not the DNA letters that control, but an orchestra of regulation performing the same score in millions of variations. Second fact: everyday habits are full of genetic traits, noticeable without a microscope. Fast or slow caffeine processing depends on the CYP1A2 gene variant. Fast metabolizers drink an evening espresso and fall asleep calmly, while slow ones experience a longer increase in blood pressure and heart rate, and, together with ADOR2 variants, feel more tremors and anxiety from the same cup. The perception of bitterness in broccoli, arugula, and tonic is linked to the TAS2R38 receptor. Supertasters perceive bitterness earlier and more intensely, more often avoid cruciferous vegetables, and involuntarily shift their diet towards milder flavors, which affects salt and sugar intake. Not all adults digest milk. Lactase persistence is determined by regulatory variants near the LCT gene. Where livestock historically developed, such variants spread, turning dairy products from children's food into an everyday resource for energy and calcium. The alcohol flush effect in part of East Asian populations is a consequence of a less active ALDH2 variant. Acetaldehyde accumulates, causing heat and tachycardia. This is a physiological stop signal that reduces the likelihood of abuse, but with persistent consumption increases risks for the esophagus. Even chronotype is marked by genes. Variants in PER3, clock, and other circadian regulators shift the internal clock, making larks more productive in the morning and owls in the evening. The same set influences the reaction to time zone changes and shift work. It is important that none of these examples rigidly dictate behavior. Slow caffeine metabolism does not prohibit coffee, nor will bitterness doom you to dislike vegetables. Tastes can be trained, doses adjusted, and schedules adapted. But genetic settings explain why the same stimulus for different people differs so much in strength and consequences. Third fact: your genes lay out the blueprint for building and reconfiguring your brain, determining when it learns most easily and how it handles stress. In the early years of life, following their plan, billions of synapses are laid down. By adolescence, some connections are pruned and pathways become myelinated. The speed and accuracy of signals increase, as if asphalt were laid over old dirt roads. Plasticity genes, those controlling growth factors and synaptic chemistry, like BDNF and its receptor, regulate how generously the brain rewards training with new connections, They determine whether vocabulary comes easily in childhood or how quickly you master a guitar skill in adulthood. The Val66 MET variant in BDNF, according to numerous studies, alters activity-dependent protein release. Some carriers have slightly poorer average memory performance and higher anxiety, not a sentence but a noticeable nuance. The COMT gene, which breaks down dopamine in the prefrontal cortex, has fast and slow variants that shift the efficiency curve differently. Some find it easier to keep tasks in mind under calm conditions, others under moderate deadline pressure. Under overload, any variant falters. Rare mutations in FOXP2 demonstrate that genetics also plays a role in articulatory motor skills. Families with speech fluency disorders and difficulties in programming speech movements have been described, not a language gene, but a conductor of several neural ensembles. Stress genes, including cortisol sensitivity regulators like FKBP5, show how experience and heredity intertwine. Difficult childhood events can alter methyl marks and the reactivity of the hypothalamic-pituitary axis, amplifying or dampening anxious responses. There are debated topics, for example around serotonin transporter polymorphisms. Science is clarifying the effects. The main point, heredity sets the ranges, and learning, sleep, exercise, psychotherapy, and environment move the sliders within these ranges, reshaping the trajectory of attention, memory, and resilience. Fourth fact, diseases are not just mutation equals disease, but a complex risk map where some genes play as soloists and others as a choir. Soloists include rare but potent variants. Mutations in BRCA1/BRCA2 significantly raise lifetime risks of breast and ovarian cancer. Knowledge of them changes screening routes and even leads to preventive surgeries. And in therapy, drugs targeting such tumors' vulnerabilities are used. Familial hypercholesterolemia, defects in LDR, APO, or PCS9, raises bad cholesterol from childhood and accelerates atherosclerosis. Here, early analyses in relatives and aggressive treatment are useful, including drugs inspired by the discovery of protective loss-of-function PCS9 variants in some people with naturally low cholesterol. There are also shadows of probability, polygenic risks, where hundreds and thousands of weak variants add up. In a small proportion of people, their sum for heart attack, diabetes, or some cancers is comparable to rare monogenic defects. And this shifts not won't happen, but the onset age and the strength of external factors needed for the disease to manifest. A special area is pharmacogenetics. Carrying HLA-B5701 predicts severe hypersensitivity to an antiviral drug. HLA-B1502 in some Asian populations is linked to the risk of dangerous skin reactions to certain anti-epileptic drugs. Hence, testing is recommended there before prescription. In everyday life, the most accessible genetic test is family history. Early heart attacks in close relatives, recurring cancers, unusually high cholesterol indicate where to get checked earlier and more often. Yet, even high hereditary risk remains a stage for choice. Diet, exercise, sleep, quitting smoking, and timely checkups noticeably shift the odds, because genes set the baseline configuration, and lifestyle the trajectory on this map. Fifth fact. Sports genetics is not a ticket to the champions and not a ban on achievements, but a set of traits that a coach can work with more precisely. The ACTN3 protein, located in the Z discs of fast muscle fibers, is not synthesized in some people due to a variant with premature termination. Such muscles behave a bit more endurance-oriented, saving phosphocreatine and holding moderate paces longer, while carriers of the working variant are more often found among sprinters and jumpers. The ACE polymorphism, associated with the activity of the angiotensin-converting enzyme, has correlated in some cohorts. The I allele is more common in endurance throwers, the D allele in strength disciplines. The effects are small, but over long distances, they provide a statistical advantage. There are weak links in connective tissue. Variants of Col5A1 and neighboring collagens alter tendon elasticity and the risk of Achilles tendinopathy. For such an athlete, it is more useful to dose plyometrics and pay attention to eccentric loads. At altitude, genes for the response to hypoxia come into play. Regulators of the HIF pathway, EPO, EPAS1. Populations that have lived for centuries in thin air have developed a more economical oxygen transport strategy, avoiding excessive blood thickening. Rare mutations in MSTN, myostatin, demonstrate a removed muscle growth limiter. But in ordinary life, growth and strength are the result of dozens of regulators and load signals. In some, training programs cause a lush hypertrophy response. In others, it is more modest, but capillarization and mitochondria grow better. Inflammatory markers like IL-6 can predispose to prolonged muscle soreness and slow recovery. And this means not impossible, but you need to be smarter. Proper distribution of intensities, sleep, protein, iron, volume control. From here arises personalization. Two runners with the same time over 5 kilometers benefit differently from the gym, intervals, or altitude training. Genetics suggests where to shift the emphasis, but the final result is still forged by kilometers and years. The sixth fact. Character traits and inclinations are also not a coin with heads and tails, but a spectrum in which heredity provides the framework and experience paints the picture. Twin studies show that for many behavioral traits, the contribution of genetic differences is noticeable but far from complete, and there are no specific courage genes or generosity genes. There are weak contextual associations. Variations in dopamine receptors, e.g. DRD4, have been linked to novelty seeking, but the effect is unstable and strongly depends on the environment and assessment methodology. Variants in the oxytocin receptor, OXTR, have appeared in studies on empathy and trust. However, the same allele in a warm, supportive family gives more subtle social sensitivity, while in a disadvantaged family it leads to vulnerability to anxiety and detachment. The famous story about MAOA and aggression is actually about the interaction with early trauma. Without an adverse environment, the genotype hardly predicts behavior and certainly does not lend itself to labels or legal conclusions. Even school success is now described by polygenic indices. They explain only part of the variation and partially reflect genetic guardianship, how parental traits create a conducive home environment for learning. Finally, stress reactivity and self-control are not statues. Epigenetic marks on regions associated with cortisol change under the influence of care, sleep patterns, psychotherapy, and regular attention training. Imagine a shy child with a sensitive genetic setting. In a noisy classroom, they freeze, but in a well-thought-out environment with gentle rules and rituals, this same child becomes a master of subtle negotiations, not despite their genes, but thanks to them, because a flexible nervous system unfolds best where it is allowed to safely train skills. Genes here are not a sentence or indulgence, but an instruction on how to handle your temperament. The more accurately it is read, the more conscious the choice of environment and habits. The genetic landscape is an archive of recent evolution and subtly guides everyday decisions through the environment's legacy. The sickle cell variant allele of beta hemoglobin in the malaria belt grants carriers an advantage against severe Plasmodium falciparum infection, although in homozygotes it results in disease. G6PD deficiency also historically limited Plasmodium's proliferation but made red blood cells vulnerable to oxidants, hence cultural prohibitions on fava beans and caution with certain medications in the Mediterranean and Asia. The practically fixed negative Duffy receptor (DARK) variant in parts of tropical Africa blocked entry for P. vivax. In Tibetans living at altitudes, regulators of the HIF-EPAS1 pathway have been selected, allowing tolerance of hypoxia without excessive blood thickening, while the Andes offer a different adaptation profile, higher hemoglobin and lung volume. The copy number of the salivary amylase gene AM1 varies. Populations with a historically starchy diet more often carry more copies and produce more enzyme, altering the speed of carbohydrate pre-digestion and the glycemia curve. Skin pigmentation is a trade-off between UV protection and vitamin D synthesis. SLC24A5, SLC45A2, OCA2/HERC2 variants are distributed by latitude and influence the risk of sunburn, skin cancer, and vitamin D deficiency in the same geography. FU2 secretory status, whether ABO blood group sugars are displayed on mucous membranes, is linked to vulnerability to a number of gut viruses. Some non-secretories are less susceptible to common norovirus strains but have a different microbiota composition. Even blood groups behave not as personality types but as biochemical keys. Associations with cholera severity and certain malaria complications have been noted in various studies, though effect strength depends on strain and circumstances. These nuances do not dictate fate, but silently suggest where diet will be more familiar, where the sun will be moderately beneficial, and where infection will require special vigilance. Eighth fact: pharmacogenetics shows how genes regulate not whether a drug works or not, but what pathway is open for it in the body. The enzyme CYP2D6 converts codeine into morphine. In rapid metabolizers, due to gene duplications, pain relief comes faster and stronger, up to the risk of overdose at usual doses. While in slow metabolizers, codeine hardly works. Therefore, the same pills produce opposite effects. The antiplatelet drug clopidogrel is a prodrug. In variants with loss of function of CYP2C19, its activation is reduced, and cardiologists opt for alternatives in several cases. Warfarin dose adjustment depends on both CYP-C9 and VKORC1. Some patients are more sensitive and require lower starting doses to avoid bleeding, while others are more resistant. Thiopurines for the treatment of leukemias and inflammatory bowel diseases rely on the enzymes TPMT and NUDT15. With low activity, toxicity increases, so tests allow for dose reduction in advance. The transporter SLC01B1 is linked to the risk of myopathy at high doses of certain statins, and UGT1A1 affects the tolerability of irinotan in oncology. These examples do not mean that the gene prohibited the drug. Often, it is about adjusting the dose, choosing the molecule, or enhanced monitoring. And this is how genetics turns from an abstraction into safety. An important detail is context. Age, concomitant medications, liver and kidney function affect pharmacokinetics no less than genes, and associations vary between populations. But the metabolism map encoded in DNA explains why the standard average dose for one person is normal, for another insignificant, and for a third too much, and why medicine is moving so actively towards therapy personalization. Ninth fact: aging is not one broken spring, but many regulators that alter the pace of repair and the economy of resources in each cell. Some genes determine how often autophagy, the cleaning of damaged proteins and mitochondria, is turned on. Others, how readily cells divide and how quickly their telephone wires, telomeres, shorten. Hereditary differences in telomerase components, TERT/TERC, set the background. Families with very short telomeres more often encounter early problems with blood formation and lungs, while excessively long ones are also not a gift. Higher probability of certain tumors. The quality of DNA repair links aging to rare accelerated syndromes. Mutations in WRN, LMNA, or related pathways cause dramatic early tissue wear and show the price of accumulated breakdowns. At the level of energy exchange, the IGF-1, mTOR, AMPK signaling nodes are important. The evolutionary trade-off, grow now or save and be durable later, is genetically tuned. And that's why in population studies, variants in FOXO3 in long livers repeatedly surface. And in lipid metabolism, changes in CETP affecting the good and bad cholesterol profile. Carrying epsilon 4 in APOE noticeably raises the risk of Alzheimer's disease and alters the brain's response to trauma and sleep. But here too, it's about probabilities, not fate. In some, epsilon 4 will remain a background until deep old age. In others, it will play out earlier when combined with external factors. Mitochondrial DNA accumulates typos over the years, and its set we receive only from our mother, so the maternal line sometimes manifests in endurance, thermoregulation, and speed of fatigue. What's important is something else. The basic genetic setting makes the organism more or less sensitive to lifestyle. The same lack of sleep for one will cost short-term inattention, for another a noticeable cognitive decline. The same diet calorie count with different lipid metabolism gene variants reflects differently on triglyceride levels. Genes don't put a date on a passport. They set ranges for the pace of wear and repair efficiency. Within these ranges, regular movement, light cycle, nutrition, and social connections, which paradoxically affect biological markers of aging no less than molecular switches, decide a lot. 10th fact. Immunity is a place where genetic uniqueness resonates particularly loudly in everyday life. Depending on the HLA variants you carry, your body knows which fragments of viruses and bacteria to show to T cells and how strong the response will be. Some HLA alleles in epidemiological studies are linked to slower progression of certain infections, others to greater vulnerability to specific complications. But the clearest picture is seen in autoimmunity. HLA-B27 dramatically increases the risk of ankylosing spondylitis, the common epitope in HLA-DRB1 rheumatoid arthritis, and HLA-DQ2-DQ8 combinations are necessary for celiac disease, where gluten becomes the spark for small intestine inflammation. However, necessary does not mean sufficient. Carriers are many, those who get sick are the minority, and environmental triggers, microbiota, and age are decisive. Skin barrier impairment due to variants in FLG, filaggrin, makes eczema and atopy more likely. Dry skin allows allergens to pass through more easily, and in some children, this triggers the atopic march towards asthma and allergic rhinitis. Genetics even affects vaccine response. Differences in HLA and innate recognition receptors, like TLR, partially explain why one person after a hepatitis B vaccine course gets high antibody titers while another needs revaccination. At the intersection of immunity and reproduction is the subtle scene of maternal KIR receptors interacting with fetal HLA-C. Their combinations statistically influence the risks of preeclampsia and fetal growth characteristics, another example of how genetic dialogue defines life paths before birth. We have already encountered FU2 as a secretor of blood groups on mucous membranes. This is a good example of how a single genetic setting changes both resistance to specific viruses and microbiotic composition, and thus the propensity for inflammatory flare-ups. The sum of these details turns into everyday life. Some find it more beneficial to thoroughly moisturize their skin and avoid harsh detergents. Others with a rich family history of autoimmune diseases pay more attention to atypical symptoms and screening. And some know that a low vaccine response is not bad immunity, but an individual setting for which there are enhancement protocols. Immune genetics does not dictate what you will get sick with, but it forms a map of probabilities and clues along which you walk every day, often without noticing the road signs, but following them.